evaluation. In the yesterday's class, we have read drug adulteration. Both these, drug adulteration and drug evaluation, both of them comes under analytical pharmacognosy. Means in this what we do, we examine the crude drugs. We examine the crude drugs regarding its quality, purity, adulterants which are present in it. So it is called as analytical pharmacognosy, analyzing the crude drug. Adulteration comes in it, and also drug evaluation also comes under analytical pharmacognosy. Coming to today's topic, that is drug evaluation. The first definition of drug evaluation. Evaluation of crude drug means confirmation of its identity, determination of its quality and purity, detection of type of adulteration. So this is the definition. In the examination, this you have to mention, because definition means the entire topic what you read in this drug evaluation all should come in 2 to 3 lines. That is called definition, right? So it all covers it. Evaluation of crude drug means confirmation of its identity, determination of its quality and purity, detection of type of adulteration. First, coming to confirmation of its identity, means from which it is been taken, from which species it is been taken, and which part it is been taken. Drug evaluation of drug means we validate a drug whether it is from the same source or different source, confirming the source whether it is from the same source. Like for example, fennel we take it from Feniculum vulgare. It is from Feniculum vulgare only. So confirmation whether it is taken from that or some other species. So that is confirmation of its identity. Next, determination of its quality and purity. The fennel what you have taken, that is of pure quality or impure form, that is standard fennel or substandard fennel, that we have to evaluate. And then detection of type of adulteration, whether the fennel is been adulterated, with what it is been adulterated, what is the adulterant present in it, that is detection of type of adulteration. So in this topic, we generally evaluate a drug, we validate a drug, we examine the drug. That is called as drug evaluation. Once again, the definition: drug all should mute your audio. Ganesh, mute your audio. So evaluation of a crude drug means confirmation of its identity, from where it is been taken, determination of its quality and purity, detection of type of adulteration. So this is the definition of drug evaluation. Then coming to what are the reasons why we perform the drug evaluation, means biochemical variation in the drug. Why we have to evaluate the crude drug means, if the drug has undergone some chemical changes or if the drug is being degraded, to check that we evaluate a drug. Biochemical variation of the drug. If there is any biochemical variation, why the biochemical variation occurs? If it is not stored properly, if it is not dried properly, in those cases the chemical constituents which will be present in the crude drugs, that will be degraded, that will be decomposed, degraded. So in order to check that, we evaluate a drug. Biochemical variation in the drug, effects of treatment and storage of the drug. Effects of treatment and storage of the drug. So if it is being dried, drug we collect from the plants, and that will be present in the fresh form containing the moisture, and after drying we require the dried form of the drug. After drying, we have to evaluate a drug, because when it is been dried, it has undergone any changes in its chemical constituents. If it is over dried, drying also, it is for each drug different conditions will be there. One will be dried at this temperature, one should be dried at this temperature. Above that temperature, that drug will be degraded. So to check that, we usually evaluate the crude drug. And last one, adulterants and substituents. So in order to check the adulterants, whether the drug is having any adulterants or not. So these are the main reasons why we evaluate a crude drug. I think all have understood what do you mean by drug evaluation. Yes, who is this? This is your second class and you are doing in this way. Who is Viresh T? Viresh K. Next time I will not admit you to the meeting if you perform all this. Okay, I think all have understood what do you mean by drug evaluation. If anyone did not understand, can ask now. All have understood. So we shall go for the next types of evaluation. What are the different types in evaluating a crude drug? We are having mainly the 5 types of evaluation. First one is organoleptic evaluation or morphological evaluation. Second one is microscopical evaluation. The third one chemical evaluation. Fourth one physical evaluation, and the fifth one is biological evaluation. Who has done this? Better organoleptic evaluation. It is also referred as morphological evaluation. Second one is microscopical evaluation. Third one is chemical. Fourth is physical, and fifth one is biological evaluation. First, First one is morphological evaluation. It is also referred as organoleptic evaluation. Organoleptic evaluation means a study of drugs using organs of senses. It refers to the methods of analysis like color, odor, taste, size, shape, special features like touch and texture of the crude drug. So in morphological evaluation, what do we do means we use our sense organs and evaluate a drug. How do we evaluate? By its morphology, by seeing its color, by seeing its odor, taste, size, shape, and special features like touch and texture. By its touch and texture of the crude drug, we evaluate a drug, whether it is a pure form of the drug, or whether it is a standard drug or substandard drug. We evaluate the drug. So the name it has come, organoleptic evaluation, by using the organs of senses. By using our sense organs, we evaluate the drug in morphological evaluation. Next, the study of this form is called as morphology, while description of the form is called as morphography. This study is called studying the external features of the crude drugs by using our sense organs is called as morphology, and what we write, the description of the drug by seeing with our senses, that is called as morphography. Morphology and morphography. Examples: how do we evaluate a drug? First, the fractured surface in cinchona, quillia, cascara, bark, and quassia wood are important diagnostic characters. Next, aromatic odor of umbelliferous fruits, sweet taste of licorice, wavy shape of Rauwolfia, pungent taste of capsicum, ginger, brown color of cinnamon, and odor and taste of spice drugs like asafoetida, black pepper, nutmeg, caraway, cumin are important diagnostic organoleptic characters. Another example, disc shape of Naxwamica, ovoid tears of acacia, ribbon shaped tears of Tragagan. So what all I have said now? What all characters I have said now? Those are called as the diagnostic characters. For that drug, it is The diagnostic character means that character is constant for it. First one, what did I say? The fractured surface in case of bark drugs. Bark drugs is cinchona, cascara, quassia wood, quelea. These are some of the bark drugs. So what do you mean by fractured surface means? Here you can observe. See, this is the so this is cinchona bark. What I have zoomed here. This is called as the cinchona bark, and at the tip. So at the tip, this is the tip of the bark. So after breaking the bark, what you get the surface that is called as the fractured surface. So fractured surface of each bark drug will be different. Some will be having a smooth fractured See, while I am explaining, your audio should be mute. Whenever I ask a question, at that time your audio should be on. So remember that. Again and again, I won't be saying all this. So this is the fractured surface. When a bark is being broken, the surface what you get, that is called as the fractured surface. Next, aromatic odor of umbelliferous fruits. Umbelliferous fruits. Can anyone give me examples of umbelliferous fruits? Akila? Chaitra? Nikhil? Yes, no one don't know what are umbelliferous fruits. Examples are fennel, coriander, all these are caraway, all these are umbelliferous fruits. So these will be having aromatic odor. Why they will be having aromatic odor means because they contain volatile oils in them. So that is a diagnostic character of umbelliferous fruits. All umbelliferous fruits should have an aromatic odor. So that is how we evaluate umbelliferous fruits. When you take an umbelliferous fruits, first thing what you evaluate is by seeing its odor, whether it has aromatic odor or not. Suppose if it has aromatic odor, then it's okay. It is of good quality. What else if it is not having aromatic odor? Means it tells us that it doesn't contain volatile oils in it. It is an exhausted drug. That is the meaning if we don't get the aromatic odor from the umbelliferous fruits. Then the next is sweet taste of licorice. Licorice, it is a drug. It is a root drug, licorice root. It will be having sweet taste. Whatever licorice from any region you take, licorice will be having sweet in taste. So this becomes its diagnostic character. To evaluate the licorice, we have to see its taste. If it is sweet in taste, then we can say that this licorice is of good quality or good standard. These are the diagnostic characters. Yes.
Ma'am, licorice means what, ma'am?
Licorice, it is a root drug which you get in glycosides, saponin glycosides. It is a root drug. So that licorice, how do you evaluate? Enಗೆ ಅದನ್ನ ನಾವು what you say evaluate ಮಾಡ್ತೀರಿ ಅದು good quality licorice, bad quality licorice means by seeing its taste. Licorice from any region you collect, from any part of the country if you collect, it should be having sweet in taste. Then only we can say that this licorice is of good quality.
Ma'am,
Right.
that is only
Next.
containing roots only, ma'am.
Yes, once again.
That is only containing roots only, ma'am.
Licorice, it is a drug, it is a plant. So from that plant, we only take the root because that will be containing the saponin glycosides. We only take the part of the plant. Which part we take? Only the root we collect, right? Kavita has
Yes,
asked me the question. Yes.
yes, ma'am. I
Did you
understood,
understand?
ma'am.
Okay.
Thank you, ma'am.
What these are all means, these are the diagnostic characters ma. Andre iv hinge irbeku. Licorice ge it should be sweet in taste. That is the way of evaluating the licorice. Then umbelliferous fruits, fennel, coriander, caraway, ajwain, these are all umbelliferous fruits. Those should be having aromatic odor. Good odor, aromatic odor they should be containing because they contain volatile oils in them. If they don't contain aromatic odor, we can there itself we can tell these are of not good quality. Next example, the wavy shape of Rauwolfia. Here you can see the Rauwolfia drug. So this is the Rauwolfia drug. Rauwolfia root. We take the root from Rauwolfia plant, Rauwolfia serpentina. Rauwolfia serpentina, we collect the root from that drug. So it is always wavy in shape. Rauwolfia, from wherever we collect Rauwolfia, it will be having wavy nature. Wavy in its shape. So it is also called as snake drug. Because of its wavy nature, it is also referred as snake drug. In Kannada, it is referred as Sarpagandha. Sarpagandha. Rauwolfia, it is called as Sarpagandha. So it is its diagnostic character, Rauwolfia. Next, pungent taste of capsicum and ginger. Whenever you eat ginger and capsicum, you feel somewhat pungency in its taste. A strong taste, you can feel it in your mouth. That is because of the chemical constituents present in it. If they don't contain these pungency in their when you take these drugs, then you can understand that it is not good quality. The quality of the crude drug is not proper. Next, brown color of cinnamon. Cinnamon, that is called dalchini. Cinnamon is dalchini, right? So that is always brown in color. It can't be any other color, right? So it is its diagnostic character. Cinnamon, it should always be brown in color. If it is light brown in color or some other color or red in color, it is not cinnamon at all. In this way, we can evaluate cinnamon. If someone has brought you cinnamon and given, some vendor has come to you and sold, uh, gave you, this is cinnamon, please buy. So how you evaluate that cinnamon? It should be brown in its color and it should have an aromatic odor because it also contains volatile oils in it. So in that way you evaluate that crude drug. Next, odor and taste of spice drugs like asafoetida, black pepper, nutmeg, caraway, cumin are some of the important diagnostic organoleptic characters. So, yes, who is Arsha? Erase it. I did not tell you to write or scribble anything on my screen. So this meeting will be recorded. Don't think that I am just taking. Again, you should be answerable. So odor and taste of spice drugs like asafoetida, black pepper, these all drugs what I have mentioned here, they will be having, these are called as the spice drugs and they will be having distinctive taste. Each of them, asafoetida, it will be having its own taste, black pepper, it will be having its own taste, nutmeg, caraway, cumin, all will be having its own taste. So by tasting these drugs, you should be able to evaluate those drugs. If they don't possess that taste, you can say that this is not black pepper. So if black pepper is been adulterated with papaya seeds, when you taste that, you can easily distinguish which is black pepper and which is papaya seeds. So that is its diagnostic character, its odor and its taste. Next, disc shape of Naxwamika. So here are the Naxwamika seeds. What I have zoomed here. So these are seeds, Naxwamika seeds, which contain indole alkaloids. Alkaloids they contain. So all Naxwamika seeds, if we take from any part of the country, those should be disc shaped. Those should not be any other shape. Those should be disc shaped. I think you all know what is disc shaped. Can anyone tell me which is the cell in our body containing disc shape?
RBC.
Yes, good. RBC's red blood cells are disc shaped. So Naxwamika seeds also should be disc shaped. If they are apart from disc shape, we can easily say that this is not Naxwamika. So disc shape is the diagnostic character of Naxwamika. Then ovoid tears of acacia. Here you can see the tears means tears are nothing but the lumps. Lumps which are acacia, it is a gum. So lumps of acacia, those should be ovoid in shape, round in shape. So that is the diagnostic character of acacia. Ribbon shaped tears of tragagant. In case of tragagant, it is also a gum. In case of tragagant, the tears are ribbon shaped, flat. So that is the diagnostic character of tragagant. So these are few examples which comes under the morphological evaluation or organoleptic evaluation. Still many examples you can give. Turmeric, yellow color of the turmeric. Still pale yellow color of quassia wood. Same way, what you use in your daily lives, the umbelliferous fruits or volatile oil containing drugs, you can take the examples of those and you can write it here in morphological evaluation. I think all have understood what is morphological evaluation. You have got a basic idea what is morphological evaluation. Shall we move on to the next type of evaluation? Next type of evaluation is microscopic evaluation. Morphological evaluation by seeing with our sense organs, we evaluate a crude drug by checking its color, odor, taste, size, shape, texture, and touch of the crude drugs. We evaluate the drugs. Then coming to microscopic evaluation. In microscopic evaluation, by the name itself you can understand, by seeing, observing it under the microscope, we evaluate the drug, right? So this method allows more detailed examination of the drug, and it can be used to identify the organized drugs by their known histological characters. So what all drugs we read it in our textbooks, those will be present in the pharmacopoeias, in the standard books. So while evaluating that drug, we can take the reference of those books and evaluate the drug. In this case, we can compare the TS. We can compare its microscopy by seeing with the standard books and with the specimen what is present in front of us. So here I have, for your understanding, I have given you one example, that is TS of Naxwamika. This is the TS of Naxwamika. Naxwamika seeds in morphological characters, you have seen Naxwamika seeds. So when you take the TS of that Naxwamika seed, it should have this characters. It should have these cells. It should contain trichomes in it. It should contain this plasmodesmatal connections, endosperm it has to contain, parenchymatous cells it has to contain. These are the compulsory cells or compulsory tissues which should be present when you take the TS of Naxvamika. Only example I have given you for your understanding. Then it is mostly used for qualitative evaluation of organized crude drugs in entire and in the powdered forms. Every plant possesses a characteristic tissue feature. So this type of evaluation, microscopic evaluation, this is performed only for the organized crude drugs. This point you have to remember. Why you can't perform with the unorganized crude drugs? Can anyone tell me? Why you can't perform with unorganized crude drugs? Already I have told you the difference between organized and unorganized crude drugs.
They didn't contain volatile oil, ma'am.
No, Kavita. Yes, Viresh. You can unmute and say. Because I will be saying organized. Yes, yes, Anita.
Unorganized means extraction of
Right. Unorganized drugs means these are not the parts of the plant. They are just an exudations or extracts which are been obtained from the plant. And in this microscopic evaluation, what we examine the tissues or the cells which are present in the drug. If cells are present in the extract, how can we examine the drug? In organized drugs only we will be having the definite cellular structures. In unorganized drugs we won't be having any cellular structures or any cells, any definite cellular structures or any arrangements of the tissues we won't be having in the unorganized drugs. Only in the organized drugs we will be having the cellular structures. And again, while taking the TS, we can take the organized drug TS. For example, leaves we can take, stem we can take, root we can take, then fruit also we can take, seed we can take. So these all by keeping it by keeping it on our hand, and we can take the TS transverse sections of that drug. Then how it is possible in case of unorganized drugs? Either those will be present in the powder form, or those will be present in the form of semi solids, or those will be present in the form of liquids. How can you take a TS of those? Heng examine ಮಾಡ್ತೀರಿ microscope ಮೇಲೆ ಹೆಂಗ್ ಇಡ್ತೀರಿ? That is not possible. So this method, microscopic evaluation, this is only meant for the organized crude drugs like stem, bark drugs, stem drugs, seed drugs, organized drugs. This is applicable only for the organized drugs. Then the organized drugs, it can be in the form of entire drug, full. For example, seed part entire drug form alla irbodu, illandre it can be in the form of its powder also. If we make powder of that drug, then the cells will be broken, right? But if we place that powder onto our slide and examine, we can see those cells, broken cells also. We can able to we can identify that this is from this drug only. This powder is cinnamon. We can identify. So this is applicable only for the organized crude drugs in its entire form and in the powdered form. Here we can evaluate the quality of the crude drug. How can we evaluate the quality of the crude drug? By seeing its chemicals. For example, let me take the volatile oils. When I take a TS of phenol, it contains volatile oils in it. Clove, it contains volatile oil in it. When I take the TS of that and observe it under the microscope, oil cells will be present. Within that oil cells, oil should be present. Oil will be yellow in color. If it is yellow in color, that you can observe it under the microscope, then you can say oil is present and it is of good quality. If the oil cells are empty when you take the TS, oil cells are empty. Simply cell is been present, inside that nothing is been present, empty cell is present. So in that case, yeah, you can say that the volatile oil is not present in the drug, and it is of poor quality drug. And you can evaluate by taking the TS of the drug, microscopic evaluation. So microscope can be used to confirm the structural details of the drug from the plant origin. Mute your audio, Muhammad Ali. Microscope. For this case, we use the microscope. Here, our sense organs cannot be used. These are in micron scale. We will be using the microscope to evaluate the structural details of the drug. Various reagents or stains can be used to distinguish the cellular effective results. Various reagents or stains can be used to distinguish the cellular structures. So in this case, while after taking the TS, we will be placing it on the microscope and observe it, and we will be observing it under the microscope. So you can't differentiate the tissues, or you can't differentiate which is the parenchyma, which is the collenchyma, which is the sclerenchyma, which are the vascular bundles in that, because all will be of the same color. You can't able to distinguish what are these cells. So for this case, we will be using some reagents in order to distinguish the cells. For example, fluoroglucinol and concentrated HCl. This is one chemical combination, and in the pharmacognosy lab also we will be using this chemicals only, fluoroglucinol and concentrated HCl. So when you add this, so which cell it will stain? It will stain lignin containing cell. Lignin is a what is lignin? Lignin is a pigment which is present in the cell wall of the cells. Right. So which all cells contain lignin in it will stain by this reagent, chloroglucinol and concentrated HCl. Here you can see few of the cells which are being stained with the lignin. They appear pink in color or red in color when they are being stained with these staining solution. These are called as the staining solutions. And similarly, mucilage is stained pink with ruthenium red. Mucilage, for example, aloe vera juice, it is a mucilage. Then agar, it is a mucilage. So when you add ruthenium red to that solution, it will stain pink. By that way, you can tell that it is agar, because agar, acacia, tragagantha, different powders will be there. So for how can you evaluate that? By this way, which contains mucilage. You will be adding for acacia ruthenium red ಆಗ್ತೀರಿ, tragagant ruthenium red you will add, agarge you will add ruthenium red. Agar, acacia and tragagant, they won't acquire pink color because they are gums. They are gums. Gums don't acquire pink color when you add ruthenium red to that solution. Only mucilages, they acquire pink color. Mucilage is stained pink with ruthenium red. Example for mucilage containing drugs is isabgol, agar, aloe. These are few mucilage containing drugs. Still another examples. Cellulose swells and dissolves in Q-oxime. What is Q-oxime? It is ammonical copper oxide. It is a reagent. What is Q-oxime? Ammonical copper oxide. Short form, it is referred as Q-oxime. Q-oxime, what is this reagent? So when you add this reagent to cellulose containing fibers, for example, cotton, it contains cellulose in it. When you add this, the cotton fibers will swell. You can see swelling of the cotton fibers when you add this Q-oxime reagent. Ammonical copper oxide. Once again, I am repeating, ammonical copper oxide, that is called as Q-oxime. When you add it to cellulose, it swells. Next, N by 50 iodine solution. This N by 50 means 0.05 normal. Iodine solution stains starch and hemicellulose blue. Starch grains and hemicellulose, if it is present in your crude drug, And when you add iodine to that solution, it will stain blue in color. Starch ge, when you add iodine, it will stain blue color. So this is one chemical to identify starch grains. Then, so this is all about the reagents which are used to distinguish the cells, which distinguish the tissues which are present, cells which are present, components which are present in the crude drug. Because when you take the section, all will be of the same color. You can't distinguish which is a starch, which is a cellulose, which is lignin, then which is collenchyma, which is parenchyma. You can't distinguish. So when you add these reagents, it will be easy for you to identify which is the cell present there. I think you have all understood. Any doubts in this? Any doubts? You got a basic idea regarding this, right? What is microscopic evaluation? You have to reply me, or else I'll be keep on saying, if you don't understand, then what is the use of it?
Yes, understood,
Yes,
ma'am.
ma'am. Understand.
Did you understand? If you want me, wherever you find difficulty, you ask me, I can explain.
Okay, ma'am.
Okay. Then let us go for some other cellular structures. Mute your audio. Some other cellular structures, that is lignified trichomes in Naxwamica, warty trichomes of senna, wavy medullary rays of cascara bark, glandular trichomes of mint. So Naxwamica, it should contain lignified trichomes only. Already I have shown you the Naxwamica picture. So when you take the TS of Naxwamica and stain it with chloroglucinol and HCl, ನಾನು ಆಗ್ಲೇ ಹೇಳಿದ್ದೀನಿ, if you take simply Naxwamica TS, add glycerin, mount it and observe it under the microscope, you can't distinguish which cells are present. So in this case, for every TS, you have to add some reagent in order to distinguish the cells. So in this case, Naxwamica is stained with chloroglucinol and HCl. After staining that, see these trichomes, these are all called as the trichomes, the pink color one, which are stained because they contain lignin in it and they acquire pink color. These are so called as lignified trichomes. So this is the diagnostic character of Naxwamica. Naxwamica andre they should contain the lignified trichomes in them. That is only the Naxwamica. Then warty trichomes of senna, some bent trichomes. Senna should contain trichomes which are bent. Then wavy medullary rays of cascara bark. The medullary rays in cascara should be wavy. They should not be straight. That is the diagnostic character of cascara. Then glandular trichomes of mint, which secretes, which contains glands. The trichomes which contains glands in them are called as the glandular trichomes. So mint leaves, they should contain glandular trichomes. It is a diagnostic character of mint. Lignified trichomes in Naxwamica, warty trichomes of senna, wavy medullary rays of cascara bark, and glandular trichomes of mint. These are the few diagnostic characters of the crude drugs. Next. Mute your audio, Shanti. Next, the powdered clothes do not contain sclerites or calcium oxalate crystals, but both of them are present in the powdered clothes stocks. While explaining the microscopic evaluation, I said you it is a qualitative evaluation. We evaluate whether the powdered cloth is present in its entire form or in the powdered form. I said you. So here they have given you one example. The powdered cloth do not contain sclerites or calcium oxalate crystals, but both of them are present in the powdered form. I said you. So here they have given you one example. The powdered cloth do not contain sclerites or calcium oxalate crystals, but both of them are present in the powdered form. I said you. So here they have given you one example. The powdered cloth do not contain sclerites If you have gone to the market and brought clove powder for your pharmacognosy lab, how do you evaluate whether it is the clove powder or some other powder, whether it is only the flower bud or along with its stalk, that is petiole, along with its petiole it is been powdered? How can you evaluate? Means here they have given you the example. The only flower bud, if they are if the powder it contains only the flower bud, it should not have sclerites or calcium oxalate crystals. Both are absent in the pure flower bud of clove flower bud. Both are absent. Sclerites, calcium oxalate crystals, both are absent. So these both are present in the clove stalks. Clove stalks alli sclerites, calcium oxalate crystals. These both are present in the clove stalks. Henge identify ಮಾಡ್ತೀರಿ? If we take that powder and examine under the microscope, if it contains calcium oxalate crystals, sclerites, then you can say that this clove is been adulterated with the clove stalk. Clove stalk kuda aite ee powder olaga. Clove flower bud jate clove stalk kuda ನೀವು powder ಮಾಡಿ ಹಾಕಿದಿರಿ. So that is the way of evaluating the clove. This is one example. Next, you will be having linear measurements and quantitative microscopy. What are these linear measurements and quantitative microscopy? Let us discuss in the tomorrow's class. What are leaf constants? What is linear measurements? What are linear measurements and what is quantitative microscopy? In the coming class, when you are having, in that class, we shall discuss. Till now, if you are having any doubts, now you can ask me. It may be any drugs. It may be any topic. See, why I am asking these many times means this is an online class. If I explain it on the board, you may have you may ask there itself. And this is okay.
Yes, ma'am. Ma'am.
Then in the tomorrow's class we shall, or the coming class when you are having see your timetable, in the class we shall discuss regarding the next topic, linear measurements and quantitative microscope.
Okay, ma'am.
Thank you.
Okay, ma'am.
Okay, ma'am.
Yeah, ma'am.